Tamam. Good morning, sir. Hello.
Good morning.
Yes, sir. I'm audible, sir.
Yes, you are.
So, okay, sir. Can we start?
Sure, sure.
Okay, sir.
Can I go ahead?
Wait, sir.
Sure.
Sir, before we are going to start, I would like to invite our resource person, Mr. Anup Abhijit Chaudhary, sir. He is M.Pharm in pharmaceutics from BPT Odisha. He is currently leading differentiated formulation research and development at Dr. Reddy's, providing direct leadership to multiple teams of highly skilled formulation and process scientists. Previously, he has worked across state-of-art research and development organizations like Dr. Reddy's and Ranbaxy. Currently, Sun Pharma. He is having more than 21 years of experience. He is having more than 21 year of experience uh, in pharmaceutical product development. Except all, he has also led different projects spanning ideation, strategy development, product development, technology transfer, registration, approval, and commercialization. He is co-inventor in 25 international patents, co-authored 20 research articles in various national and international journals, and co-authored two book chapters for a reputed publishing house. Sir has represented Dr. Reddy's in various international and national conferences, delivered seminars, and presented posters. Mentored several graduates, postgraduate, and PhD students from premier educational institutions like NIPER, BITS, IIT, BHUs, etc. for their dissertation work. Current area of research include bioavailability enhancement, phosphate variability reduction, novel drug delivery system, gastroenteric drug delivery system, long-acting oral solid and liquid dosage form, oral peptide delivery, etc. And he is currently having his designation as Vice President and Delivery Manager, Differentiated Formulation, Research and Development, Dr. Reddy's Lab. So we are welcoming Anup sir, and we are very much delighted to have him as a resource person, and hope all our students will get a lot of knowledge from his experience, what he is going to share. Thank you, sir. If
Thank
there
you.
is director, okay, director sir is not present now. He will join in between. So, sir, please, sir.
Thank you, Dr. Simanchal, for introducing me to the team. And hi, everyone. So, without delaying further, I will just start my presentation. So, today's topic is differentiated formulation. So, I'm not sure that how much familiar you all guys are with differentiated formulation. I will start from the basics, like what is differentiated formulation, and what are those activities or the development we are doing in the differentiated formulation. And in the presentation, I will try to cover that what are the different aspects of differentiated formulation, what technologies we are involved in differentiated formulation development. So before we start, actually, I just wanted to take you guys through the one visual representation. So if you so I hope you guys are able to see the visual representation, which is a pharmaceutical product risk reward matrix. And in the x-axis, it is a risk of failure, and in the y-axis, you can see that it is a reward with respect to the the revenue what an organization may expect out of a pharmaceutical product development, and also the investment risk, like in case of failures, what is the investment risk. So with that, if you see, so the extreme right corner, the NCs comes. So here the risk is very high, investments are very high, but in case of success, the rewards are also very high. So as you are aware that for developing NC molecule, more than $1 billion dollar is required. More than actually 10,000 molecules. Only one, if you screen more than 10,000 molecules, probably you will get one or two molecules or lead candidate for further screening. And probably by the time actually we develop the NC and bring it to the market, it takes about 10 to 15 years of time. So this is a very high reward, high risk development. At the same time, if you see in the extreme left corner, the generics comes. The generics, when I'm talking about, these are actually generic for the US
Sir,
market. Yeah,
sir, just a second.
sure.
Sir, now our directors will join.
Yeah,
So I would just like to invite our director, sir,
sure.
to invite you for expert talks.
Sure.
Director, sir.
Good morning, Dr. Anup.
Good morning, sir.
Nice to be here, and though it is, I joined late,
No worries.
so I will listen your uh, what you call presentation. Then I will get back to you. Please
Sure. Thank you. Thank you. So, as I was telling that if you can see the extreme left corner, these are generic formulations. When I'm talking about generic formulations, these are generic formulations for the US, Europe, Canada, kind of like advanced market where the rewards are actually comparatively lower compared to NCs. At the same time, the risk of development, risk of failure is also very low. So these are extreme opposite. Like NCs and generics are completely opposite. NCs are new molecules, new chemical entities, but generics are actually old chemical entities, but a copy version of the innovator formulation or NC formulations. But when we are talking about differentiated formulation, it comes in between the NC and generics. So differentiated formulations, if you see, are moderate risk. At the same time, actually, it has moderate rewards also. Certain times it is actually there are some differentiated formulations which are highly differentiated product. So that there you will get a lot of clinical benefits. So your rewards are also going to be very high, but at the same time there are low differentiated product which I will be taking you through what is actually exactly differentiated product is about, and there actually the rewards will be low. At the same time the risk is also low, so it comes in between NC and generics. And in the same space you can see also biosimilars. So biosimilars or biologic formulations also comes very near to the differentiated formulation with respect to the risk versus reward. So before going further, I just wanted to actually tell that if I can give a definition of differentiated formulation. So differentiated formulations are the formulations which addresses any unmet patient need. So unmet patient need, we'll go further and we'll understand what is unmet patient need. So generally, just to keep you in mind, like what is unmet patient need? These are actually the lacunas in the current gold standard of the formulation or current innovator formulations. What are the lacunas in that? So any formulations which are actually addressing one or more unmet patient needs through formulation intervention using advanced drug delivery system. These are called differentiated formulations. So we will go further and understand what are actually unmet patient needs. So as I told, unmet patient needs are actually the gaps in the current medications or the current gold standard of the medications. So if I can divide the unmet patient need into broad categories, then it can be categorized into three broad categories. One is lack of efficacy, second is lack of safety, and third one is lack of patient compliance. So lack of efficacy is that, for example, when a product is taken, current product is taken, not a wide population gets kind of like efficacy, and they get actually cured from that disease. Lack of safety, there could be some side effects or severe side effects, serious side effects associated with drug molecules. So those are actually kind of like contributing towards the lack of safety. And lack of patient compliance, there are products like which is available in the injectable form. So it is very difficult for a patient that if you give a formulation in terms in the form of injectable, which has to be taken for a chronic disease, where the formulation has to be taken or the medication has to be taken for a longer period of time, and probably every day or multiple times a day. So that will be very difficult if it is given in the form of embryo. Just I'm giving some example with respect to that. So we will go further and understand deeper all this, what are lack of efficacy, lack of safety, and lack of patient compliance. And I have some case studies also, at least one case study I will take you through with respect to all three tenets of the unmet patient needs. So going forward, so I just trying to emphasize and elaborate on what is lack of efficacy. So lack of efficacy could be due to majorly poor bioavailability, inadequate drug concentration at the right time, inability to inability of the standalone drug or a single drug to provide the adequate action. Again, if you see poor viability can be due to different reasons. So we will go through each one of them. So poor viability could be due to inadequate solubility. So how we are going to resolve that? Generally, solubility is a intrinsic character or inherent character of a molecule, but it can be improved through formulation and process interventions. Like if we can develop the product like a amorphous solid dispersion, and amorphous solid dispersion can be made through different routes, like hot melt extrusion, spray drying, spray granulation, and there are new technique like electrospinning, electrospraying. Then there are new technology like in the solvent precipitation technology or solvent free precipitation technology, like where we can use supercritical fluid technology to make amorphous solid dispersion. Similarly, if you see, we can also reduce the particle size. We can micronize through the jet mill, we can pulverize, we can make nano size through the bead mill, or we can use high pressure homogenizer to reduce the particle size. So what will happen when you reduce the particle size? When you reduce the particle size, the overall surface area is increased kind of like geometrically, and with the increase in the surface area, overall surface area, the wettability also increases. Also, what happens actually, your exposure to kind of like the dissolution or solubility has increases. So in that way also we can improve the solubility, where the poor viability is due to the inadequate solubility. There are some drugs which follows pH dependent solubility. You might be aware that there are certain drugs which are only soluble in the acidic pH. There are drugs which are only soluble in the basic pH. So for that, what we can do based on their area of absorption or site of absorption. For example, one drug is actually solubilized only in the acidic condition, but not in the basic condition, but it absorbs from the kind of like lower part of the intestine. Then what we can do is that we can actually use alkalizers and modify the microenvironment of the gut so that the pH kind of like increases or decreases based on that. Like for example, we want to increase the acidic solubility. So what you will use? You will use acidifiers to reduce the pH and solubilize the API, so that in the intestine the pH decreases and the drug gets solubilized and gets absorbed, because the site of absorption is lower part of the intestine. Similarly, if some drugs are actually dissolved in the alkalized condition, so we can also create alkaline condition using some alkalizers. Then next, coming to poor viability due to lack of absorption. So if there is a lack of absorption. So what we can do is actually mainly I'm talking about the drugs which are actually class 4 molecules, class 3 molecules. So where actually your solubility and permeability can be a rate limiting. So permeability in case of class 4, like that Kate is a completely impermeable drugs. So what we can use is actually we can use permeation enhancers for that, or we can also use targets, targeted release to the specific part. Like uh, they are actually some drugs are there actually they are very kind of like target specific or really site specific absorption it happens. So for example, there are drugs which are only absorbed through either through the stomach or in the upper part of the intestine. So what we can do is that we can use gastroretentive drug delivery system. Gastroretentive drug delivery system can be made into different part like floating, mucoadhesive, or we can actually use like gas generating gastroretentive drug delivery system. And if in case the drug is having a site specific absorption from the lower part of the intestine like colon, we can also do a colon targeting. So we using adequate coatings or the coatings which are only opening in the higher pH. Like when we are targeting colon, we should actually target like more than 6.5 or 7 pH, where actually the when the drug will reach the colon, there only it will dissolve and it will kind of like release the drug from the formulation. So in that way, we can actually target the colon. So similarly, the poor viability could be, as I told, that it's an intrinsic property. So it could be also due to the particular salt or particular form of the drug. So what we can do is that we can also develop new salts, new prodrugs, so that it can help in overall absorption of the drug. uh, then we can also understand like in case the poor viability is due to the in vivo drug degradation, like we know that proton pump inhibitors, and there are other drugs which are actually very pH dependent degradation, like they are not stable in the acidic pH. So then what we can do is that we can use a enteric coating or pH dependent polymeric coating. Then what we can do is that we can bring up with a formulation which will only open when it reaches to a particular alkaline pH, maybe, or alkaline, or at least higher acidic pH, like maybe 5.5, 6, 6.5 pH, so that it would have already crossed the stomach by that time. And that's how actually we can prevent the degradation from the stomach. So it is mainly entry coating. And there are products like, for example, there are some cardiac drugs which should be actually in the peak concentration at the early morning time. So what we have to do is that actually if a patient takes the formulation in the evening, then what will happen, the drug will immediately release, and it will give the peak concentration or the Cmax maybe near to actually kind of like when the patient is going to the bedtime. But the drug should be available in the right concentration during the early morning. So what we can do is that we can do chronotherapeutics. Chronotherapeutics means we can do a time release. We can give a gap. The patient has to take the formulation in before going to the bed at 10 o'clock. So we will give a coating in such a way that the coating will dissolve in maybe 6 to 7 hours. So for example, if a patient is taking the formulation at 10 o'clock in the night, then after 6 hours, maybe at 4 o'clock, it will start releasing, and it will be the drug will be observed within an hour time. So at the 5 o'clock in the morning, it is in the highest concentration where actually the drug is required in the desired concentration. So in that way also we can improve the efficacy. Similarly, if you see inability to of standalone drug to actually provide adequate, like that there are certain drugs actually like paracetamol. Today, if you see, if you have a fever, certain times paracetamol works, certain times paracetamol alone doesn't work. So what we do actually, we add another drug to that. Like for example, we have drug with paracetamol plus mefenamic acid. So it works synergistically, and it is a synergistic fixed dose combination, and it gives a better efficacy. So I just want to keep a pause over here and try to understand from the team that if any questions are there.
Dr. Simanchal?
Please go ahead, sir.
Okay, great. Thank you. So then kind of like then we'll go for lack of safety. So what is mean by lack of safety? Like lack of safety could be kind of like adverse side effects. So adverse side effects could be local side effects, it could be systemic side effects, then it could be kind of like a due to a particular API, due to it is a particular route of administration. So we'll go through each and everything. Like for example, there is a local side effects. So what you can do, like for example, there are drug which you take in concentration, it gives a lot of irritation in the GI tract. But or let us consider that if it is a topical formulation, it's a cream, lotion, or ointment. When it is given in a particular concentration, gives a lot of irritation to the skin. But when how can we actually improve that? So if we can reduce the overall dose, but increase the permeability, so that or increase the viability so that the local side effects can be reduced but without impacting the overall efficacy, because we are not impacting the systemic exposure or the systemic absorption. So if the systemic absorption is not getting impacted, only the local absorption is getting local kind of like concentration is getting reduced. So we are targeting both. We are reducing the adverse effect due to local concentration. At the same time, we are not compromising on the overall efficacy. Similarly, you know that there are products which are very narrow therapeutic index, or there are products which are actually Cmax related side effects will be there. So what we can make sustained release kind of formulation. So we can do the pharmacokinetic modification for developing like a modified release formulation or a sustained release formulation where we can blunt the Cmax. The Cmax can be designed in such a way that the Cmax will be well within the therapeutic window, so that there is no impact on the overall efficacy. At the same time, we can reduce the Cmax related side effects. Similarly, if we see that there are drugs, like for example, we know that when we take PPIs, the proton pump inhibitor, or let us consider that we take NSAIDs. Okay, so when we take NSAIDs, we know that like for example, diclofenac, aceclofenac, or nimesulide, they are having gastric side effects. We get a lot of gastric irritation. So what we do generally, we take antacids, or we take proton pump inhibitors, we take H2 antagonists. So with that, so that it counters the general side effects which are actually majorly due to the kind of like gastric irritation or kind of like ulcerations. So when we combine a NSAID with either a PPI or either a H2 antagonist or any antacid, we come up with a synergistic FDC, so that there will be no such side effect. All and there are multiple other products, like you know that drug has effect, also a side effect. So there are products which are actually those are drugs which counters the side effects of other drug. So what we can do is that we can bring those two drugs together, and we can come up with a synergistic FDC or fixed dose combination, so that one drug counters the side effect of others. By that way, actually we are actually countering the side effect of each other, and we are not getting any side effects because of this combination. Then coming to adverse effect because of the route of administration, you know that there are certain injection sites. Like for example, there are some oily injections. When we actually give this oily injection, we get an injection site irritation, pain, inflammation. So what we can do is actually we can convert those injectable to oral, or oral to topical. So in that way, if we can reduce, change the route of administration, we can actually come out of this adverse effect, which is specific to this route of administration. So next I will go to the lack of patient compliance. So the patient compliance could be due to multiple reasons. Like one is high pill burden. So when it is high pill burden, it could be frequent dosing, like if we tell the patient to take every 6 hours or every 8 hours, the patient has to take 4 times a day, 3 times a day. And for example, there are, that in this kind of like age where actually there are a lot of lifestyle diseases like diabetes, hypertension. People are actually patients are there actually who are taking number of pills. I know that I have seen people who are taking daily at least 10 to 12 tablets, and that too in different timing. So what we can do is that if we can come up with actually reduce the dose by biology enhancement, so that what will happen that instead of actually taking multiple pills, they can take one pill. Or if we can actually develop it as a modified release formulation, what we can do is that instead of taking 3 times a day, 4 times a day, or 2 times a day, they can convert we can convert it into once a day, so that we can improve the patient compliance, and we can improve the dosing compliance also. Most of the people actually in the geriatric patients, they forget to take the medication on time, and that's why they see that most of the time actually they complain that the drugs are not working. So in order to improve that, we can actually come up with actually formulation which can be given in kind of like a single dose, or it can be given once a day. So in that way actually we can improve this patient compliance and dosing compliance. Second thing is that that there are drugs which has to be taken before food, which are to be taken after food. There are drugs which are to be taken absolutely in the fasting condition. There are drugs which are to be taken during the food. So there are food restriction, and the food restriction is because there is a pharmacokinetic change happens. Like for example, I'll tell you there are multiple oncomolecules. They have kind of like different solubility in different food. Like for example, if your food is low fat versus high fat, the solubility of the drug will be different in the food, and that's how the overall solubility, absorption, and viability will be differing. So we have seen many drugs which are mainly oncomolecules, or the molecules for cancer has been labeled very in bold, and in black box warnings are there, actually should not be taken with food. Because what happens actually, if the patient takes with food, and the absorption happens more, then it will create some toxic conditions. So for that, what we can do is that we can improve the pharmacokinetic modification. We can through improve the formulation through pharmacokinetic modification, and we can make the formulation in such a way that it can be taken without regards to food. So this can be done. One of the ways actually we can do it through kind of like through a new salt, new polymorph, maybe improving the viability, improving the solubility. So through that actually we can also improve the food restriction. Then route of administration. You know that there are route of administration like injectable, rectal. These are not very kind of like conventional route of administration, and the patients are not very comfortable in actually taking the medication through these routes. So what we can do is that if we can develop formulation from injectable to oral, or rectal to oral, so then probably actually we can actually improve the patient compliance. And nowadays, lot of actually swallowability concern, fluid restrictions are coming because of the age factor and lifestyle diseases. Like for example, geriatric and pediatric patients, they face lot of problem challenges with respect to swallowing the oral formulations. And there are certain disease conditions, maybe there are some kidney conditions, there are some heart conditions where fluid restriction is there. Fluid restriction are actually it is actually limited to kind of like certain patient pool, so that their kind of like their overall fluid is actually restricted to maybe 100 ml or 200 ml for hour or something like that, so that they face a lot of problem in actually swallowing the drug. So what we can do is that we can also bring small change like ODT, the oral disintegrating tablet, mouth dissolving tablet, sublingual tablet, so that they don't have to actually kind of like crush the tablet. They don't have to actually kind of like swallow the tablet as whole, but just they have to keep it in the mouth, and the drug will kind of like disintegrate, and it can be easily swallowed. At the same time, there are new diseases like coming, like that GERD and other gastrointestinal diseases is coming, where actually the there is this patient also faces a challenge with respect to dysphagia. Dysphagia means they face problem with respect to swallowing. So developing formulation as ODT, MDT, and sublingual can also help that kind of patient pool also. So these are overall with respect to the different unmet need and how we actually kind of like resolve each unmet need through all these formulation intervention. Now I will take you through the case study for each one of them. But I just wanted to give a pause. If there is a question, I will try to answer. Are there any questions
Yes,
or
sir.
can I move on?
We can, sir. Can a food be synergistic in nature? If it is, how can we modify the formulations?
Can you come again? Sorry.
Yes, sir. I'm asking that whenever we are consuming the food,
Yes.
sometime that may give some synergistic effect to certain kind of drugs. And if such, so how we can modify different type of formulations?
Okay. So, if I understood correctly, that you are asking like if food has impact with respect to solubility and absorption, then how can we modify it? Is my understanding right?
Right.
Okay. So what we can do is like, for example, I can give an example where we have seen that as I was talking about an oncomolecule, where the oncomolecule is restricted only in the fasting condition. It is told that it should not be taken in the fed condition, because what happens actually, the drug has different solubility at different kind of like the food, because the different food has different fatty condition. So even high fat meal, low fat meal. So what we do actually, we develop a formulation in such a way that overall solubility of the molecule we increase to a such a extent, so that there will be no effect with respect to food. Like for example, the challenge happens because the drug's inherent property is that it is having low solubility at low fat meal, and high solubility in high fat meal. But if I can change the overall solubility to a high using maybe different prodrugs, different salts, or converting them to amorphous solid dispersion, and I can make them kind of like solubility, kind of like food independent. Then what I can do is that I can reduce the overall dose of the drug, so that if you compare the formulation in fasting condition and fed condition, it will be same. Is Did you understood what I'm trying to say?
Yes, sir. Yes, sir. We got
Okay.
this.
Okay. Thank
Thank you,
you.
sir.
Is there any other question?
We can proceed, sir.
Thank you. So I will take you through the some case studies. So my first case study is actually addressing lack of efficacy. So here I'm giving an example of actually how we have done the biology enhancement of itraconazole. So I'm just giving some examples which are actually available in the public domain. So here what I'm trying to say is that hope you guys are aware that itraconazole is a antifungal molecule, and nowadays like antibiotics, lot of resistance is there. So now fungals antifungals are also started showing a lot of resistance. So maybe in near future we are going to face a lot of challenge, because what is happening, because of multiple reason, people have started actually gaining resistance to itraconazole, kind of fungal resistance. So what is the reason? Actually, if you see, we have seen that most of the formulation which are available in the current market, they are varying with respect to viability. So even in general, the drug has 57% viability. And we have seen that because it's a poorly soluble drug and it exhibits pH dependent solubility. In general, the itraconazole is highly soluble in acidic pH and low soluble in basic pH. But challenge is the major absorption of itraconazole happens in the intestinal region, or mainly with respect to lower part of the intestinal region. So what happens actually when it goes to stomach in the conventional dosage form, whatever drug it releases and gets dissolved. But the challenge is that it has a lower absorption from the stomach and upper part of the intestine. So when it reaches to the lower part of the intestine, all the soluble portions get actually precipitated and excreted unchanged, or excreted in the feces without getting absorbed. And that's why the overall solubility, overall viability of this molecule is 57%. And if the formulation is not designed properly, then the viability further reduces. Like we have seen in Indian market, there are products available, itraconazole products available, they have zero viability. So it is shocking, but we have seen that it is having zero viability. So we should be very careful actually. If we are not getting kind of like any efficacy, we should actually contact our medical caregiver or the doctor, and explain to them that I have been taking this medication from so and so date, but it is not effective. So they can understand because whether it is working or not. So what has happened actually, I'll tell you that I'll take you through this formulation. Like when the product was initially developed, itraconazole formulation is developed by Janssen and Janssen. What they did actually, it's a BCS class molecule and it is sparingly soluble in water. So if you can see, what they did actually, they have come up with a formulation which is a pellet based formulation, pellet filled into capsule. And what they used is actually they have used a HPMC polymer based formulation. As you know that HPMC is a pH independent polymer. So that what happens actually when we develop a formulation of itraconazole with HPMC, it doesn't show any pH dependent solubility. It is only independent of pH. So what they did actually, the Janssen and Janssen formulation came with a capsule and they have come up with a pellet which is coated or which is with HPMC. So what has happened actually, this is a gold standard in the formulation since 1992. And what we have seen is that the viability is only 55 to 57%. And because the drug gets dissolved in stomach and when it goes to the intestine, it gets precipitated. And that's why only 55% is absorbed and rest of the 45% gets excreted like unchanged. Then in actually, if you see, there is a company called Mine Pharma, which is from Australia. What they did actually, they just changed the HPMC to HPMC phthalate, which is a pH dependent polymer. And when you see pH dependent polymer, so it is actually pH dependent polymers actually, it is only opens in the higher pH. So this formulation, what they did actually, they have come up with two formulation. One is Logenac, which is 50 mg. So 50 mg is equivalent to 100 mg of formulation, and they came up with the 65 mg. 65 mg into 2. So it is 130 mg is equivalent to 200 mg. So how they did? So they came up with the SPMC phthalate, and they come up with a process of spray drying, so that the drug, when it is taken, because it is pH dependent and SPMC phthalate doesn't open in stomach, when the drug goes into intestine, it starts dissolving. And as I told you in the beginning that itraconazole is kind of like having a higher permeability in the lower part of the intestine, And because it's a amorphous solid dispersion, due to HPMC phthalate, the overall solubility of the formulation is also increased in the higher pH. So we have given both conducive environment to the itraconazole now, where we have dissolved the drug in the higher pH. At the same time, in the intestine, the drug is absorbed well. And that's how what they have seen is that they have actually seen a more than 100% viability enhancement in this formulation. So you can see, and they have given it as a supra-bioavailable technology or SUBA technology. So if you see this formulation, they have what they did is actually they did a viability comparative viability study, Lozanac 50 mg versus Voronax 100 mg. Although they could not get 100%, they could get around 77 to 80%. But what they have done is actually they have seen that AUC by MIC ratio they have checked, and they have seen that in all concentration from 4 to 16 microgram per ml, they have seen that MIC kind of like MIC 90 is from 4 microgram per ml to 16 microgram ml. In all condition, it has actually shown superior or similar efficacy. So through that, actually, they have got the approval in Europe and Australia. But when they went to USFDA told that, okay, it is fine, but you do a bioequivalent formulation. So what they did, instead of 50 mg, they came up with 65 mg. And 65 into 2 is 130 mg. They did a bioavailability or bioequivalence with 200 mg of formulation. Now you can see that they have got a formulation which is near to 100%. So they have two formulations. So currently, it is, yeah. Any questions?
अरे चाल दी क्लास चाल दी
No, sir, no.
Okay, great. So they have a formulation which is actually 50 mg formulation is equivalent to 100 mg of Sporanax, and 130 mg of formulation is equivalent to 200 mg of Sporanax. So this is one of the example. So next case study is actually addressing the lack of safety. So where I am going to talk about clobetasol propionate, as you are aware that it is a formulation which is actually used for actually mainly the psoriatic conditions and also other skin conditions. So like it is actually the innovator is Temovate, which is clobetasol propionate cream, 0.05%. These are steroids, these are corticosteroids, and you know that corticosteroids are having lot of systemic side effects. It actually suppresses the pituitary axis and which has actually direct correlation with respect to our immunity. So what we have seen is that when Temovate is actually launched initially, they have actually Temovate is the innovator. It is the concentration is 0.05%. This is the strength. Okay. And you can just have a look into the what are the ingredients they have used. But when we developed a formulation, what they used to happen actually, it used to give the efficacy, but the challenge is actually it used to also have a lot of kind of like impact with respect to pituitary thalamus axis or pituitary axis, which is actually impacting on the overall immunity. So what we did actually, we come up with a formulation. We just slightly change the formulation. We choose the right permeation enhancer. So you can see these two bold or the ingredient which is given in a different font, diethylene glycol monoethyl ether and isopropyl myristate, IPM and transcutal. This is nothing but transcutal. These two, when we used, what we did is actually we could come up with a formulation with the half strength. Instead of 0.05, we can see that we can come up with a 0.025% formulation. And you can see we did comparative study. You can see the plasma concentration. So when we did our 0.025%, our plasma concentration is only 56 picogram per ml, and whereas the Temovate formulation is 152.5. So you can see that 1/3 reduction with respect to the systemic concentration, which reduced our incident of hypothalamus pituitary axis suppression, adrenal axis suppression from 36.4 to around 11%. So you can consider that it has 1 by 4 reduction in the HPA axis suppression, and so that we could actually see a lot of reduction in the side effect. At the same time, if you see the efficacy, our formulation, when we did we did the two studies against actually vehicle, the placebo. So you can see in two studies we got a separation of 15% and 14%, which showed a significant. As you can see, the p-value is less than 0.001. Both the study actually showed very good kind of like separation, and you can see the efficacy through this uh, pictures, where you can see that, see, this is the baseline or the initial, when you can see the day 10, and you can see day 3, day 10, and day 15. So you can see that after day 15, almost the plaque psoriasis has actually completely removed. Okay. And at the same time, you can see in this case also in the study 2, where actually we can also see that you can see after day 15, it is completely removed. Okay, so there are two different people. Like it is a 53-year-old woman. It is a 26-year-old woman. So you can see that it has completely reduced. At the same time, you can see that there are visual evidence with respect to the efficacy, how the product was, how the disease was before the medication and after medication. You can see this is before medication and after medication after 15 days. So we can actually could actually come up with a formulation. And you know that corticosteroids are actually restricted to only maximum use of 15 days. But with this formulation, what we have done is actually we could see that there is no impact even if we are using up to 30 days. Like for example, anybody uses beyond 15 days also, it is not going to impact much. Like third case study is actually addressing the lack of patient compliance, where actually I'm just showing that how innovator has actually initially actually developed tofacitinib formulation as a immediate release formulation, which is actually a 5 mg formulation, and how they have actually come up with a formulation with 11 mg extended release formulation. This formulation is actually for rheumatoid arthritis, and you know that rheumatoid arthritis is a chronic disease where the patient has to take the formulation 2 times a day if it is a immediate release formulation, and to be taken for at least 3 to 4 months at a time. Certain times it is taken throughout your life, because rheumatoid arthritis is actually a old age disease, and now it is actually become a lifestyle disease also. So that's how actually it is taken for a longer period of time. So you can see here, initially the innovator Zelzan, which is from Pfizer, they come up with actually a immediate release formulation 2 times a day. Then later, what they did actually, they come up with a formulation which is XR formulation, 11 mg OD or QD. So how they did it, see, you can see this solid lines shows that actually when 5 mg 2 times is given, the graph shows like that in the in vivo, in the bioequivalence. And they come up with a formulation which is actually a osmotic drug delivery system, which they told us extrudable core technology. And through this, what they could do is actually they come up with a 11 mg formulation, which is giving equal exposure as of 5 mg 2 times a day. You can see that when 5 mg 2 times it is given, it is in a single time, it is giving 6 plus 6 hours, so 12 hours of relief. So in a XR formulation also, they could also get more than 12 hours. It is around 12 to 14 hours of release, because what they did is actually they have seen the concentration above minimum effective concentration, and they have seen is that they are getting equal or better efficacy when kind of like they are developing it as a extended release formulation. At the same time, actually they are addressing the unmet patient need of actually the patient compliance, where the patient doesn't have to take it 2 times a day, they have to only take once a day. So they have done some background backup pilot studies, or the PK studies for that. So you can see the first A and B shows that the study, when it is actually taken in kind of like single dose followed by a multiple dose study. So you can see it is like it is a single dose study and it is a 5 day study. So they have shown the bioequivalence in both conditions. You can see 100 near to 100 all places they have seen. And Cmax is to it is good that Cmax is lesser than the IR, because in the XR formulation we don't have to meet the Cmax. Cmax has to be lesser than IR, because what we are doing is that we are actually blunting the Cmax to in such a level that the Cmax is always in between the therapeutic window. So you can see A and B depicts single dose and multiple dose study. In both the cases, AUC was near to 100. And when they also did a food effect study. Their formulation, XR formulation, when they have given in fasting and fed condition, this also gave a kind of like near to 100% with respect to AUC. So with these studies, they got the kind of like FDA approval. So this is all about what I wanted to actually cover. Sorry. Sorry. So I wanted to cover in my presentation. So now I will pause and I will try to take questions, if any.
Thank you very much, sir. Thank you very much for your precious time. Sir, I just want to know, like different MDT dots are there, multiple dose are given for different disorder as
Before
antibiotic.
that, kindly,
By differential formulations, can we overcome this type of antibiotic resistant, or can we modify the formulation in single dosage form?
Yes. So for antibiotic resistance, what we should do is actually first thing, in general, antibiotic resistance, I never actually recommend to develop it as a modified release formulation, because there are formulations which are actually concentration driven, like which are diseases are concentration driven. Like for example, when you develop any antifungal, anti kind of like viral, antibiotic molecules as sustainability, there is always a possibility of kind of like developing resistance. So I will not suggest to develop it as a sustainability. Rather, what we should do is that we should see that how can we improve the viability of the molecule, so that with a lower concentration we get the similar viability. So that should be a focus for reducing the antibiotic resistance.
Thank you, sir. Thank you for your precious time. I would like to invite our director, sir, Dr. Simanchal Panda, sir, Director, School of Pharmaceutical Sciences, Jaipur National University, to tell something. Please, sir.
Dr. Anup, can I ask you for this removing the sharing of slides so that we can?
Sure. I will.
Yeah, because we need a big screen of this.
Sure.
Nice, you have presented really the need of the hour, and actually the formulators look out how to design a formulation and how to optimize the formulations, how to uh, absorb the exact dosage, what is the desired for the patient, and with less side effects, more efficacy. It's really good presentation, and the presentation what is presented over here, it is worthy, and I request if future time permits you, so we will also call and invite you for some other topics of your
Sure.
domain expertise.
Sure.
So we will be in touch up with you, sir, for our PhD panel, and also for external viva and all these activities.
Sure.
And second thing is that in your institute, say for example, any coordinated research is there, then we will collaborate with you with the university. We will exchange our thoughts.
Sure.
Yeah,
Sure.
these are the outcome of our coordinations between you and our institute. And nice, and I hope the audience is gathered here online, as well as you have also arranged a screen for them to get a combined class of you. So we hope it will much benefit for them. And we will get back to you. We'll touch with you, and these are the things. And thanks for your time giving us.
Thank you, Dr. Subransu, for your kind words. We will definitely be open for any collaboration in future. I would connect with you and Dr. Simanchal for understanding more on your current work areas and your expertise. Also, we will see that if you can help us in such kind of like solving any formulation related challenge or a design related drug design related challenge in future, and we will connect and we will see that what are your current work areas, so that we can collaborate, and I'll be open for actually any specific kind of like collaboration or any contribution towards educating your students and fellow kind of scientists. So I'll be always open. So please feel free to contact me.
Yeah, yeah. And secondly, I would tell you the School of Pharmaceutical Sciences and the Jaipur National University is having all the programs for the Pharmacy Council, starting from diploma to PhD.
Oh, great. Okay.
So, PharmD, PB, and PharmD people, they are also doing research. The pharma students are also engaged in research, and then pharma are there, PhD scholars are there. We have sophisticated labs, and infrastructure is very well. So we hope that you are tuned for the research activity. We will upheld, and we keep it up in next time onwards. I
Sure,
hope.
sure. Sure. I'll be always open, and definitely I can also help you from my organization to actually kind of like if you need any kind of collaboration, or you want any suggestions, we'll be always open for that.
Yeah, yeah. The exchange of thoughts we will do.
Sure. We'll do that.
So nice, sir. Thank you. Bye bye.
Thank you. Thanks a lot. Thank Dr. Simanjal.
Thank you, Dr.
Thank you
Simanchal.
very much.
We may leave now.
Okay, sir.
Okay. Thank you.
Thank you.
Okay, thank you very much, sir. Anup, sir.
Thank you. Thanks a lot.